All right. Hi, everyone. Happy half a week away from the AP exam. I can already tell because we have lots of questions that stress levels are high. So hopefully tonight I can bring your stress levels down a little bit by going over some of the stuff that you guys are asking about. Let me just get my PowerPoint ready. There's the dog. You saw her head. She likes to come sessions with me. It's great. Okay. Woo. My internet must be lagging. Hi, Tiffany. Welcome back. So happy you're here. Oh my goodness, look at all these questions. Okay, perfect. Well, I see that some of these questions are things that I actually went over like last week. So I may have to postpone them for a little bit just because we went through them last week. So you can watch my replay because I talked all about the nervous system last week. So again, if I have time, I will totally cover it, I promise. But I really do want to get through ecology because a lot of people have been asking for assistance with that. I had some requests last week as well. So I'm just gonna, instead of just rambling, I'm gonna go ahead and get started. Reminder, this is the last free session. So if you want it's a 1 hour for whoever's asking. If you want my Cram sessions, I'm doing a Cram session on Friday, Saturday, and Sunday, and I can post the times in here, and you can join me for those as well, and I can go through a lot more questions, but those you do have to be a 5able Plus membership, which is $5 per month, so not too bad. Okay, here we go. So I'm going to try to zoom through this. Again, just so that I can answer a lot of your other questions as well. So bear with me, but if you have a question that is pertinent to what I am talking about on the screen, maybe just ask it in the chat box so that I can kind of differentiate what questions we want to go over after this and what questions I need to answer in the moment. Okay, here we go. So with ecology, you need to know a little bit about the scope of ecology. And so that's going to range from individual all the way up through the biosphere. So you can see this here. Big differences that you need to know about are just population versus community ecology. So population is a group in the same area of the same species versus community ecology, which is going to be about different populations coexisting in the same area. I'm seeing we already have questions about what this is about. So yes, Savannah, thanks for answering that. So we're going over ecology because I have been presenting for the past year, I guess, on different topics, and my reviews, this is my 6th review. So if you're interested in going over all the other topics, I've probably reviewed them in reviews 1 through 5, and you can do that by watching my replays. Tonight, I'll talk about ecology, potentially go over a little bit with evolution because a lot of people were asking me about it last week, and then most importantly, try to answer all these questions that you guys have. But I definitely want to make sure that I cover some of the things that I haven't yet because I have promised the students this. And then if you watch my replays, you will have a consistent library of information. Okay, perfect. Very, very briefly, abiotic versus biotic, you need to know it. Abiotic is not alive, never has been. Biotic is alive or has been alive. So wood, if you sit at a wooden desk, that's actually a biotic factor because it was once living as part of a tree. So just a brief overview with that. Just wanted to show you guys this again. For people who have been asking, this is going to be potentially on your grid ends, which I saw that someone else asked about. This is a direct snip from the, what's the word that I'm looking for? I've lost my train of thought now thinking about all these things. Oh, I'm sorry, from the equation sheet that you'll get. So if you're not familiar, there is an equation sheet that you get for multiple choice and for free response. It's the same equation sheet. You can legitimately look it up. If you want to, you can Google AP Biology equation sheet just to familiarize yourself with where the information is located if you haven't done so already or practiced in your classroom. So these are some of the things that you need to be familiar with for population questions on the grid ends. So you'll see everything is defined for you. So honestly, these are really, really easy. I'm not going to go through any examples because I think they speak for themselves. But population growth, we're going to measure as births minus deaths. And then we'll talk a little bit about the difference between exponential and logistic growth in a second. So if you're looking at exponential growth is unrestricted, okay? Meaning the population can grow forever and ever and ever and ever and ever. This never really happens because there are restrictions to every population, but a lot of times we will see portions of exponential growth within a logistic growth, which is going to be way more common, which is what you see on the right. And logistic growth forms that nice S-curve because there's a carrying capacity in the environment. And a carrying capacity could be anything that's going to limit unrestricted growth. So think space, think food, think water, think any of those things, air, oxygen, anything that might limit growth is going to serve as a carrying capacity. So we're going to see that logistic growth in the middle there, but then we're going to start to slow down once we reach that carrying capacity. And if we go back just one slide, there is a different equation depending on whether you're looking at growth rates for exponential versus logistic growth. You'll see with the logistic growth equation, you're actually going to have to plug in the carrying capacity of the environment in order to answer those questions. Okay, so that's a carrying capacity, which is good vocabulary, as well as exponential versus logistic. Okay, cool. All right. Other quick vocab that sometimes teachers don't cover that I think is really, really important is the difference between density-dependent and density-independent change or factors. So there's a lot of vocabulary that goes along with ecology, which is why I'm flipping through this. I know I'm talking really fast, but I see that you guys have a lot of other questions, so I want to try to manage our time well. So density-dependent population change, so factors that are biotic would be things like predation, inter and intra-specific competition, accumulation of wastes, diseases, parasites. All of those things are going to depend on the size of the population, okay? The amount of waste that accumulates, the amount of food that's available, the predators in the area, disease, all of those are going to affect populations differently depending on the size of the population. So that's what you want to think for density dependent. They depend on the density or the size of the population. There are also density independent factors. Those types of things would be like weather. It doesn't matter if we have a population of 10 or 10 million, weather is going to affect them the same way. Temperature will affect them the same way. Fires, natural disasters, and the lovely human activity, because we are destroying our planet, are all things that would be affecting a population regardless of its size. Okay, so just something good to keep in mind. Yes, only biotic things will be dependent, and abiotic things are independent. All right, community ecology. Okay, so I'm segueing a little bit, and now we're talking about how organisms depend on one another. Okay, so you need to be familiar with ecological pyramids. So this is an example, and the main reason you need to be familiar with them is because you need to know that energy is lost. About 90% of energy is lost, and only 10% is transferred between trophic levels, which are the striations that you see along this ecological pyramid. So you'll see that producers start with 100% energy, then we decrease down to 10%, 1%, and 0.1% energy. I actually was just looking at a grid-in question with my students that looked at an ecological pyramid, and you had to determine the amount of energy that was transferred from one level to the other. So what you needed to do was look at the amount of energy in this level and divide it by the amount of energy in this level to get the percent. So 10% transferred and 90% loss is our general rule of thumb, but different ecosystems may vary slightly there. So if you see a problem like that, you can always just set up a ratio to find the percent that is transferred. Okay. Cool. So community relationships. I saw someone asked this over here. This would be all different types of symbiosis. So symbiosis is going to focus on community relationships. So I'll go through the majority of those. So we have parasitism, which is usually the easiest for everyone to remember. It's going to be a relationship in which one species benefits and one species is harmed. So you can see here we have a tick, we have some sort of gross mite on a caterpillar, and we have a mosquito. So all of these things are relationships, again, in which one relationship is benefiting, or one organism is benefiting because they're getting nutrients, a food source, a safe place to live, whatever it may be from the other one, and the other one is being harmed in the process by losing nutrients or a safe place or anything like that in return. That's usually a pretty easy one for people to remember because we all know about parasites because they're gross and fun to think about. Commensalism is kind of like the middle child, the forgotten one. Commensalism is a relationship in which one organism benefits and one organism is not harmed or helped. So we have a specific type of fish that will school around these sharks. The sharks get no benefit from having these fish near them, but the fish are going to be able to get the remnants of the shark's meal. Sharks are messy eaters, so they're going to pick up the remains of whatever the shark is killing and get a free meal out of it, but this is not affecting the shark at all. My other favorite example is barnacles. Barnacles can actually grow on the side of a whale. The whale is not harmed by this. They're also not helped by this, right? But they just grow onto the thick skin of the whale. And as the whale moves through the water, more water is going to run past that barnacle. And so the barnacle is going to get a larger amount of food than they would if they were growing on a rock that's not moving. Because again, they just have more water flowing across them, so they're going to pick up more particulate. So that's my favorite example of a commensalist relationship. Mutualism is also usually easier to remember. It's mutual, so hopefully all of your relationships are mutualistic. It's going to be a relationship in which both organisms benefit. So we have pollination with the bees and flowers. Flowers benefit because they get pollinated, and we get a lot of genetic variation. For those evolution fans out there, genetic variation is the key to life. If you see genetic variation in an answer choice on the multiple choice section of your exam on Monday, and you don't have a whole lot of time left, you should absolutely choose that answer. We also have oxpeckers on the back of a zebra. For any fans who played March Mammal Madness this year, the oxpecker and the zebra. Oxpecker picks bugs off the zebra. Zebra does not have those parasites anymore. And then anemone and clownfish, everyone's favorite from Nemo. So again, those are going to be relationships in which both benefit. All right. We also have predators and prey. Predator is going to be an organism that preys upon others, and prey is an organism that is preyed upon by others. Makes a lot of sense. Just remember that preys can also be predators, and predators can also be prey, depending on where they fit into a food web. So that is not their single role unless they are an apex predator, like a lion, in which case they do not have any natural predators. They are the top dog. All right, so I wanted to show you a few examples because a lot of people have been asking for examples of multiple choice questions. So I have a few here for you that I really want to go through. What's the difference between parasitism and predator-prey? That's a great question. Predator-prey, one of them is dying, and one of them is surviving, and it's like this hunt that happens. Parasitism is just an organism that is growing and exists. They don't want to kill their host because then they would die. They're depending on the host for nutrients, so they're not going to kill their host. They're just going to deplete it of nutrients. Oftentimes the host will die eventually, but it's a long process, so it's not their intent to kill their host, whereas with predator and prey, it is their intent. Sarah, great question. I did go over that in, I think, review 4, so maybe check out that replay if I don't have time to answer it today. All right. Cool, so let's take a look at this multiple choice question really quick. So they're ecologically based, and then it looks like we have a wide variety of other questions, so I probably won't go into evolution because I already have in review part 1 that you can replay, and that way I can focus on just the questions of the people who are in the room tonight. If there are evolution questions, though, I'd be more than happy to answer them. Okay, so I like to point out this question because there's a lot that can be learned from it. It says, one model of a sustainable fisheries practice is for individual fish to be removed from a natural population at a rate equal to the highest possible growth rate of an ideal population. The graph above represents the population of bluefin tuna, at which point in the graph is the population growth rate the highest. So a lot of my students like to pick 2 or 1. They like to pick A or B for this problem, and both are wrong. So anytime you see rates, you need to think slope, change over time, okay? So Y over X, population size over time. So the answer here is going to be C, because that's where we have the steepest slope in this graph. So I'm just highlighting that because rates show up all the time on the AP exam, and students often tend to miss that or don't know how to calculate a rate. A rate is always going to be slope. I want you to get overly confused, though. Sometimes they will just ask you to list the change, like the change in the population. That is just going to be a difference of the y-axis points, but a rate, you're always going to be doing slope. Okay. All right, next, I like this graph because I wanted to highlight this graph. I'm more interested in highlighting the graph than actually answering the question. This is your typical predator-prey graph. You need to be familiar with it. You're probably going to see it on Monday. So you'll always notice that prey has a higher population size than predator, and because of that, they usually have their own axis. So we oftentimes have double y-axis graphs here because prey is just a very, very much larger population than predator. Our populations get smaller and smaller as we move up the trophic levels. So there's always going to be less like apex predators than there are primary consumers. So that's something to note. And then you'll also see that these curves are kind of chasing one another. That's indicative of a predator-prey. You'll see when predators are the highest right here, prey is the lowest. And when prey is the lowest, when predators are the lowest, prey is the highest. And that should make sense because when you don't have a lot of predators, you're going to have a lot of prey. When you don't have a lot of prey, you're not. When you don't have a lot of prey, you probably have a lot of predators eating them. But eventually, then they obviously will not have enough food, and that's why their population decreases. All right, we can skip over this one because I know people are eager to jump into other things, but I did want to look at question 6 because this one gave my students a lot of trouble, and so I would like to go through it with you to make sure that you are confident. Look at all these answer choices. You guys are great. In a species of cactus, the number of spines on a plant is genetically determined. The graph above shows frequency distributions for populations of the cactus species growing in the presence or absence of 2 herbivores. Which of the following best accounts for the differences between them? So I'm going to let you guys take a look at this for like 30 seconds, cast your votes, and then I will go through it with you briefly. Hopefully it's not too challenging. I'll be right back also. I think my dog's getting into trouble. You read that question. Ooh, I see some different answer choices. Okay. Hopefully a lot of people have gotten a chance to read through. Okay, so our best answer choice here is B. Reason being, we need to account for the fact that we are losing both our small numbered spined and our large numbered spined plants. So we can see this here. This is an example of stabilizing selection. So there's your little evolution review. But we're losing these low number spined plants and these high number spined plants when both herbivores are present. Again, we're moving towards the center. This is stabilizing selection. So you need to pick an answer choice that shows both the loss of those two. So B is telling us that peccaries eat cacti with smaller numbers of spikes, hence why we're losing those, and that wasp larvae eat cacti with greater numbers of spikes, hence why we're losing those, and pushing towards this middle stabilizing selection. Okay, perfect. That one was just a little tricky for some of my students, so I wanted to throw it up here just to talk through it with you guys and do a little bit more practice. Okay, so it looks like the move is lots of different questions. So, let's see, where to begin? 22 questions. Holy schmoly. All right, I guess I'll stop at the top. I start at the top and work my way down and do my very darndest to try to get through all of this. I may need to pause occasionally to pull up PowerPoints and things that we have gone through so that you can take a peek at them as we go. Okay, so I'm going to pull up my PowerPoint from last week. I'm going to go through this very, very briefly, only because you can absolutely watch the replay and get a much more thorough explanation here. And I want to make sure that I'm able to answer everyone's. So here we go. All right, so I'm going to talk about an action potential because it sounds like that's what you're asking about. So let me get my action potential slide up real quick. Boom, 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 boom. All right, here we go. Things that you need to know about an action potential, first and foremost, is you need to know that the neuron is a salty banana. And what I mean by that is that it has sodium on the outside and potassium on the inside at rest. It's also, it has a lot of anions, some negative charges in the inside. You can see that here. We have a very negative interior and a very positive exterior. That actually gives us a resting membrane potential of negative 70 millivolts, okay? With sodium on the outside, salty, potassium on the inside, banana, okay? But it does not stay this way forever because we have the action potential. So basically what happens here, action potential is like dominoes. Once we reach a threshold of positivity, it goes, and it goes, and it goes, and it goes. How does it reach this threshold? Great question. Usually a neurotransmitter stimulates a channel protein. I think I have an image of that. Here we go. So here's a neurotransmitter stimulating a receptor protein. And when that happens, the receptor protein opens up, and the channel that is opening up is the sodium channels. Okay, so once we reach this initial threshold where we're like, oh yeah, this is real and not a joke, we go through this process of depolarization. So to be polarized means to be like unequal. Like if you think of like polarizing opinions, you're on opposite ends. So very, very negative. When we depolarize, we get rid of that change. We get rid of that negativity. And the reason that this is happening is because our sodium channels are open and our potassium channels are closed. So when sodium channels are open, here's our sodium channels, you will notice that just like every other molecule on Earth, sodium will move from high concentration to low concentration. So sodium wants to spread the heck out and get inside that cell and away from all the sodium on the outside. When it does that, it's bringing that positive charge with it, and we see a major influx in the positivity in the inside of the axon because of all those positive charges entering because the sodium channel is opened. So that's our depolarization moment. Now, after a certain amount of time, those sodium channels start to close and potassium channels start to open. They're a little bit late to the game. They're slow to the gate, but they will eventually open. And what you will notice here is when potassium channels open, potassium will leave because it's a salty banana. So there's lots of potassium on the inside. It wants to get out. So it's leaving, it's leaving, it's leaving, and with it, so is its positivity. So now we are repolarizing the membrane. We are getting more negative. We are getting back down to where we were. You'll notice we have this little undershoot here. And that's because potassium channels take a while to close, okay? And so we let a little bit too much potassium leave, but that's a good thing because it makes sure that we don't head back on up to the dendrite as opposed to moving in one unilateral direction towards the end of the axon and the synaptic terminals. So that's what that little undershoot is, and it is very important so that we don't send signals back up the other way. And then we got to reset. We have to reset this darn thing. And so to do that, we use the sodium-potassium pump. The sodium-potassium pump is our life's blood. It's what allows us to reset our dominoes after they've all fallen down. And it does this by pumping sodium back out to where it was and potassium back in. Now, you will quickly realize that that is against the concentration gradient. Sodium out, potassium in. That's not where it wants to go. You're shoving it back into its crowded domain. So what that means is the sodium-potassium pump uses a lot of ATP. That's an example of active transport. While these channels are wide open and they're just letting things in and out when they're open, sodium-potassium pump is able to force things against their concentration gradient and therefore requires energy to do so. Yeah, so the numbers, 3 sodium and 2 potassium, that's lovely. You would not have to know that on an AP exam, but it's good that you do. Yeah, you don't need to know any specifics like that. Like, don't waste your time memorizing specifics like that. You just need to know overall processes and why they occur. So don't stress too much because that's not what you're going to be tested on. Yeah. And so this propagates down the neuron. So you'll see the positive sodium followed by the negative potassium and then being reset by the sodium-potassium pump. And then it ends with an influx of calcium, which is leading to neurotransmitters being released, which is going to allow this thing to start again at its target cell. Maybe it's another neuron. In this case, it's a muscle cell, actually. It's going to lead to that muscle contracting because acetylcholine is the neurotransmitter for that. But that's pretty much the action potential in a nutshell. All right. You do not need to know any exact numbers for cellular respiration. You do not need to know any numbers, I don't think, at all. And maybe I'll regret saying that because I'll think of a number that you actually do need to know, but I can't think of one right now. So you can use that as your quick fix. Okay, Max is asking about Krebs cycle and Calvin cycle and how they're related. They are not related, except for that they are both cycles, which means that things start and end and continue on forever. Krebs is during cellular respiration. The big thing that you need to know about Krebs, so what happens here is we have 2 pyruvate molecules from the breaking of glucose during glycolysis. And those pyruvate molecules get broken down a little bit further into something we call acetyl-CoA. And that acetyl-CoA gets broken down even further into carbon dioxide. And carbon dioxide is our toxic byproduct. Please do not think that our oxygen becomes carbon dioxide, because that's a very common misconception, and it's not true. The glucose is what turns into carbon dioxide, and you can actually radioactively label glucose and then eat it, and then you'll breathe out radioactive carbon dioxide. Super cool, but it's an easy way to track your carbon. And the main purpose, the main thing you should remember about the Krebs cycle is that it produces electron carriers. So we're breaking bonds. Anytime we break bonds, we're releasing electrons. We also make a little bit of ATP, but not a whole lot. The main thing is that we're producing a ton of electron carriers, which is going to make us lots and lots of ATP in the electron transport chain. And that does happen in the mitochondria. Calvin cycle is going to be our energy consuming reaction in photosynthesis. It's the second reaction after the light dependent reactions. So light dependent reactions provide us with lots of ATP and electron carriers. And then Calvin cycle uses those ATP and electron carriers to convert carbon dioxide into glucose, or G3P, glyceraldehyde 3-phosphate, which is a precursor to glucose and starch and things like that. So big things, like kind of more specifically, if you're like really striving for a 5, carbon dioxide is going to go through a process called carbon fixation, which is where it's taken out of the air and made into an organic compound. And that's done by an enzyme called rubisco, or ribulose bisphosphate carboxylase, if you want to be real fancy. Then we're going to use all of those electron carriers, because think about it, electrons are used to make bonds, and we need to make a lot of bonds if we're converting carbon dioxide into glucose. So we're going to use electron carriers, and we're going to use ATP provided from the light reactions. And in return, we're going to go ahead and recycle that ATP, or the ADP, and the empty electron carriers, and we're going to send them back to the light reactions. That's actually a really important thing to remember that some people don't think about, is that not only does the Calvin cycle make glucose, which is obviously very important because then the plant and us can break it down during cellular respiration, but it's also recycling electron carriers for the plant, which is essential, which is what we get during the electron transport chain, but also why a lot of organisms go through fermentation to recycle those electron carriers. Okay. Ooh, the 4 big ideas and grid-in questions. Sure. Okay, so I don't know specifically what details you want me to go into about the 4 big ideas, because that would be like reviewing all of AP Bio content. What I will say is you can take a look. Let me actually, okay, I'm going to share something with you that you've probably not looked at, because why would you? Because you don't teach this course. There it is. And it is the entire course description that your teacher follows to make this course. Now, like, don't go scrolling through and being like, my teacher never taught me that. Like, no teacher could ever teach you all of this. It's too much. It's way too much. But it does go through all of the big ideas, and it does go through everything that you should know about all those big ideas. And it's like 80 pages long, but you can go through and take a look at everything that you are supposed to know. So I'm kind of going to table, I don't know, the big ideas question. I mean, big idea 1 is all about evolution, and the fact that evolution drives diversity in life. So that's going to be really, really heavy on like genetic variation, and how genetic variation leads to diversity, and how without variation, we don't have anything for natural selection or evolution to act upon, right? Like if we were all genetically identical, we wouldn't see natural selection occurring, because natural selection always has to act upon existing things. So mutations never just like arise in the moment, they're pre-existing, and then they just survive and reproduce better. So that's going to be the big one there. Big idea 2 is all about free energy. So this is going to be a lot with like cellular respiration, photosynthesis, biochemistry, and talking about how all of these things are allowing us to maintain our homeostasis. So it'll talk a little bit about body systems as well. Big idea 3 talks about living systems and how they store, retrieve, transmit, and respond to information. So this is a lot with cell signaling, which is honestly a really, really big part of the AP exam these days. It's heavy topic. So I would look through Big Idea 3 because I've seen a lot come out of that recently. And then 4 talks about how systems interact. So that's going to be a lot with like ecology and looking at how organisms interact with one another. But I would highly suggest it's a clickable PDF, so you can go right to the page number that you're looking for, and it actually outlines everything that's covered there in really, really great detail. So you can make this kind of like your study guide cheat sheet. Okay, cool. Ria, great question. They're all in a document if you're a 500 plus member. I can definitely go through cell signaling as well in bits and pieces for sure. Okay, so grid-in questions. Here is my advice for you for grid-in questions. So I'll just go through it broadly, and then I can post a Word document. Maybe I'll try to upload it. Yes, I'm bad. I only use Word. I don't use Google Docs. I hate Google Docs, but I'll make you guys a Google Doc. I can post some practice problems that I went over with my students earlier today, actually, funny enough, because we're on the last stretch. So here are my strategies for grid-in questions. They're at the end, right? But if you've taken the SAT before or anything like that, you know that where you can just use your brain. You can't guess on those questions. You can't guess a random number. So my suggestion is always that you go to them first. Now, what I will say from looking at 10 bajillion grid-in questions, and that is a factually accurate statement in my mind, there's always going to be like 3 easy ones, 3 simple algebra, 3, like I can show you this example of one with a cladogram. There's always going to be 3 easy ones. And then there's always going to be 3 that are like impossible. So one of the ones on the worksheet that I worked on with students today asks you to learn a whole new equation. You don't have time to learn a whole new equation on your AP exam. Absolutely not. Nobody's got time for that. So with questions like that, you're going to skip them. So again, my advice is this. Look for the 3 easy questions. Answer them first because that's free points. Then go back to the regular multiple choice. Okay? You don't want to miss out on that. If you have time at the end, you can tackle the 3 hard questions. What I will say to you, and I mean this from the bottom of my heart and from many years of trials and tribulations, and from telling my students the same thing day in and day out, just because you can answer a question doesn't mean you necessarily should answer that question. Because I'm sure, and you know, I teach stubborn students, you're AP kids, you think you can get it all right, and you can, but not necessarily in 90 minutes, okay? So yes, you can probably sit and stare at that grid-in question and get it, but it's gonna take you like 7 minutes, and that's 7 minutes that you're not gonna get back, and then you just missed like 6 easy multiple choice points. So you kind of have to take a step back and really regulate yourself and not get too caught up in any one thing, because it'll kill you, you know, it'll take you down. All right, really quick, I'm trying to find, where the heck is my math practice? Where did I hide it? Math. What in the world? You know when you just can't find the thing you want and you have limited time to find it and now you're stressed? That's how I feel right now. Let's see, maybe it's in, when you like teach this course over and over again, sometimes it's hidden in a different year. And you're like, why did I do that? Ugh. Okay, last try. Yay, found it. Everyone, good news. I'm just gonna convert it really quick to a Google document and I'll post it for you. You guys can take a look at it. Woohoo! Hey, Alexandra, great question. I also talked about this last week. So I'll go over it very briefly. Let me find my PowerPoint slide. You don't need to know a whole lot about second messengers in CAMP other than they exist, and their purpose is to serve as the signal transduction pathway between the signal and the response. Okay, so what you need to know about second messengers is that they're basically just members of a relay race team, and they pass the baton to one another from who gets first tagged because of the stimulus, and then who ends up producing the response. Okay, how do they pass the baton? They pass phosphates. So you've probably heard of kinases before. Kinases are going to be enzymes that transfer phosphate groups. So second messengers in CAMP are going to rely on kinases, or sometimes they are kinases that are just passing phosphate groups along. Phosphate groups activate things oftentimes, like think ATP, that third phosphate group is very energy rich. So those phosphate groups, as they get passed from second messenger to second messenger, CAMP being one example of that, cyclic AMP, as they get passed, they're moving along the lines, again, just like a relay race, until finally one certain protein, usually something like a transcriptase that's going to go ahead and transcribe a gene to turn it into a protein, is activated by that last second messenger, and then they will go ahead and transcribe that gene and make that protein that was originally coded for. So that's really what you need to know about those. All right, all right, all right. Okay, sorry, I got distracted and then I didn't make this into a Word document or a Google document, and I want to because these are all really good practice problems. And they show you like the wide range of gridding questions that you could be asked, I think is highly important. So let's go ahead and go in here. You can hear the inner ramblings of my brain as I go through this, but I think it'll be really helpful for you guys once I get it together in like a minute. All right. Ooh. Also, if you guys feel like your brain is just functioning at a low level right now, I totally get that. Review season is rough. So no worries there. Okay. Here we go. Grid and question. I'm going to start answering this next question while I'm waiting. All right. Cool. All right. Independent assortment. Oh my gosh, this is so funny. These are a bunch of questions that I answered last week. Yes, I can. This is very easy. All right. Independent assortment, incomplete dominance, etc. Okay, complete dominance. It is completely dominant. So all of these three things, all they have to do with is what happens to the heterozygote, right? We know that when you have two of this allele, you're this. We know when you have two of this allele, you're this. The only difference is what happens if you have one of each. So with complete dominance, when you have one of each, one is dominant and you're that thing. So let's talk red and blue. I have a method to my madness here. So if we have red and blue flowers, and you have two red alleles, then, or two, we'll call it dominant, two dominant alleles, you're red. You have two recessive alleles, you're blue. You have a dominant and a recessive allele, you're red, okay? So anything that you know about regular Mendelian genetics is complete dominance. Incomplete dominance means that neither one is dominant. Think of what it means to be incomplete, right? So neither the red allele or the blue allele is dominant. What that means is neither is going to be present in the heterozygote, okay? So instead of having red, red, and blue, we would have red, purple, and blue. So the heterozygote would be a mix between both alleles, okay? Both are incompletely dominant, neither is showing up in the heterozygote. Lastly, codominance, they're both dominant. So they are going to share that responsibility in the heterozygote, and they're both going to show up in the heterozygote. So if we had red and blue, the mix of the two would be a red and blue spotted or striped or whatever flower. Every other petal is red or blue. But both alleles are showing up in the offspring. Independent assortment just means that each time we divide, so homologous chromosomes, so we have one version of chromosome 1 and another version of chromosome 1, they could be lined up like this, or they could be lined up like this, and they can separate independently. So mom's version of chromosome 1 might go to the left, or mom's version of chromosome 1 might go to the right, and it doesn't matter. There's a 50-50 chance that each version of each chromosome is going to end up on each side of the cell. I'm not going to go a whole lot more into that because I do go into detail about it last week and in my genetics unit where I talk about meiosis and mitosis as well. But basically, it leads to a whole ton of genetic variation. For reference, we have 23 chromosomes, 2 versions of each. 2 to the 23rd is 8 million. So there's 8 million different possible egg or sperm that one individual can make just because of independent assortment. But it's going to be the same situation as law of segregation. Okay. All right, oxidative phosphorylation. So oxidative phosphorylation is what happens at the end of the electron transport chain. And it's going to be the fate, basically is what happens in the electron transport chain, the final step of cellular respiration where we make all that ATP. So oxidative because oxygen is involved. You must remember that oxygen is the final electron acceptor. What that means is, this More on that in a second. What that means is it's the final electron acceptor. This is the reason why we breathe. Oxygen is going to be our most electronegative atom that we have. And so it is going to be very, very, very hungry for electrons. So it is the final electron acceptor, and it is going to be the force behind pulling the electron through the electron transport chain. Finally, the electron and oxygen will meet up, long-lost lovers, and oxygen will be converted into water. Okay, so again, oxygen and carbon dioxide do not exist in tandem here. Okay, here we go. Sorry, I'm seeing like so many things on the right corner of the chat box and it's distracting me. I'm really easily distracted. Okay, back to the point at hand. While that is happening, so that's where the oxidative part comes in, we're also going to do some phosphorylation. Phosphorylation just means to add a phosphate group. So while those electrons are being moved through the electron transport chain, hydrogen ions are being pumped across the membrane to the outside. And this is a form of active transport, except instead of using ATP, we're harnessing the power of that electron to pump those hydrogen ions out to the outside where they're already very crowded. The end result of those hydrogens, that concentration gradient, is that they come rushing through an enzyme called ATP synthase that synthesizes ATP. So these hydrogen ions are running through from a high concentration to a low concentration. And when they do that, ATP synthase is able to provide enough energy to phosphorylate ADP, adenosine diphosphate, into ATP, adenosine triphosphate. So that is oxidative phosphorylation. That's where oxygen comes in. That's where phosphorylation comes in. I do have a whole cellular respiration screencast, like an hour long, that goes into all the details of this if you want further information. That was an example of incomplete dominance, by the way, not codominance. Codominance would be like a red and pink, a red and white spotted flower. All right, Allison, great questions. I can certainly go through Hardy-Weinberg in a second. I just definitely want to pull some slides up for that. Surface area to volume ratio, all you need to know is the larger the surface area to volume ratio, and think about it's just surface area divided by volume. That's all that calculation is, surface area to volume. The larger it is, the better, which is why cells are so small. Because if you think about it, more volume means more energy to keep that thing alive, and more, so we don't want that. We want low volume. And we want high surface area because the surface area of the cell, the plasma membrane, is where all the magic happens. It's where everything gets transferred, oxygen, carbon dioxide. It's where enzymes live, proteins are embedded there. Everything happens in the plasma membrane. So we need lots and lots of surface area with not a lot of volume to keep things alive. So again, highest surface area to volume ratio is going to be good to go. Biomass is just going to be a ratio as well. So you're just going to do the percent or the amount of something over the total amount, but it's really just a percentage or a ratio calculation. Hardy-Weinberg, I can definitely get to in a second. I just want to pull up a few slides. Yeah, give me 1 second and I'll get that. Cool. Sorry, I got to go back into the depths here. All right, where was that? That was like one of my first podcasts. It's probably terrible. Don't watch it. All right, let's see. Natural selection and microevolution. Should be in here, I think. Holy schmoly, I have so many tabs open right now, which always stresses me out. All right. Sorry guys, my internet is being like super slow. I think it's because I'm streaming, so everything's taking an inordinate amount of time, but I'm glad that you guys are entertaining yourselves by answering each other's questions, so thank you for that. Gosh darn it, this is not the one I wanted. Ooh, you know what? It's because I did a whole Hardy-Weinberg podcast. Look at that. Okay. So there's a whole Hardy-Weinberg podcast waiting for you if you want to view it. All right, here we go. Let's pull this up. All right. So what do you need to know about Hardy-Weinberg? Well, you need to know that in order for it to happen, nothing can be changing in that population. So more than just like memorizing the different Hardy-Weinbergs, like things that need to be true, just think, would this population have a lot of change in it? If the answer is yes, then it's not in Hardy-Weinberg equilibrium. Remember, equilibrium means like stable, equal, right? So you want something like a large population with no immigration and emigration, right? But instead of just memorizing those things, you can just use your common sense and think, okay, this population is probably going to be maintaining pretty steady genes. Okay, some other things that I want to point out. Well, here's our equations. We know this. They're given to you. P and Q are defined for you. All of those things are on the equation sheet, so you do not need to memorize them. What you do need to remember is that P squared is equal to the frequency of the dominant genotype. Just think about it, right? Those are two P alleles, so I always think P as in powerful. So P squared is 2 powerful alleles, so that's homozygous dominant. 2PQ is going to be 1 powerful and 1 quiet allele, so that's heterozygous. And then Q squared is 2 quiet alleles, so that's homozygous recessive. The big thing that you need to know about Hardy-Weinberg that students oftentimes get tripped up on, let me see if I have an example. Okay, so in this practice problem, it says in a certain flock of sheep, 4% of the population has black wool and 96% of the sheep have white wool. If black wool is recessive, what percent of the population is heterozygous? So the first thing that you need to realize, one, recessive does not always mean the lower percentage. So don't get tripped up. Like, make sure you're reading carefully, because sometimes like 60% of the population could be recessive, and that's okay. Two, you always need to, if they're giving you the genotype, which in this case they are, or they're giving you phenotypes actually, but one of them is a genotype. You always need to use the recessive phenotype, because the recessive phenotype equals the recessive genotype. Because if you look recessive, it's because you're homozygous recessive. You can do absolutely nothing with that number that says 96% that's tied to the dominant phenotype, because that is a mix of homozygous dominant and heterozygous. And I say this 10 billion times, and my students will always put 0.96 is equal to P, or 0.96 is equal to P squared. One, you can't set anything equal to P or Q unless it explicitly says allele. If it said 96% of the alleles are white, then I could set 0.96 equal to P. Okay? But unless it says allele, if they're talking about whole organisms, they're talking about phenotypes and genotypes. Okay? Two, I can't use that 96% equal to P squared, because you don't know that those 96% of sheep are homozygous dominant. You know nothing about their genes. They just look white, but they could be heterozygous. So don't forget about the fact that this is going to be a mix of homozygous dominant and heterozygous. So you cannot set it equal to P squared. Please don't do that. It kills my heart. Okay, so if you know that you need to start with the recessive phenotype 10 times out of 10, you are going to get the right answer every single time, because the math is not hard. The hard thing is figuring out what the given is. So again, knowing that 0.4 is equal to Q squared, sorry, 0.04 is equal to Q squared, we can square root that to get Q, and then we can use Q to find P, because P plus Q equals 1, and then we have P and Q, and we can solve for the heterozygote. Cool. So that's my review of Hardy-Weinberg, but you can certainly watch my whole review if you want more information than that. All right. Oh my gosh, so many questions, so little time. Wow. I hope you're all coming to Cram sessions. Oh my gosh, thank you for the Cram sessions. You're so welcome. That's going to be, actually, let me tell you when they are. Okay. So it's going to be Friday. I'm going to type this into the chat box too. Friday. It is going to be, sorry, I'm in Central Time. I'm from Chicago. Which is 7 p.m. Yes. Is that right? Yes. Let me check the schedule. Just so I can get information so that you can all show up and be there on time. Okay. Sorry, no, I didn't lie to you. Yes, it's 6 p.m. Central, 7 p.m. Eastern, which would be 4 p.m. Pacific. Sorry. Time. Okay, Saturday. It is going to be 5 p.m. Central, so that's 6 p.m. Eastern or 3 p.m. Pacific. And then lastly, Sunday is the same as There it is for you. All right, Gibbs free energy. Wow, Gibbs free energy. You do not need to worry about Gibbs free energy. I cannot remember the last time that I saw one. If you do see it, it'll be one grid-in question, in which case it's not worth your brain space. You got to start thinking that way. It's an 80% to get a 5. So even if you're shooting for a 5, you can get 20% of the questions wrong. Gibbs free energy, my biggest suggestion to you is just look at the equation sheet. So I can post that really quick. But it's truly not worth your brain space to stress about it. Can't remember the last time I saw it. Okay, this question, so I already reviewed ecology. Behavior, hopefully we talked about that as well, mixed in with ecology. If I haven't, you can re-ask your question. Common ancestry. So common ancestry is just when two organisms had their most recent common ancestor. And the more closely related you are to an organism, the more recent your common ancestor was. A really, really big misconception that students have, like people think that humans evolved from monkeys, and we didn't, because monkeys still exist, and common ancestors do not exist anymore because we diverged, right? Divergent evolution from that common ancestor. So if we diverged from monkeys, or if we came from monkeys, then monkeys would no longer exist. We just have a very recent common ancestor that we've diverged from. Everybody has a common ancestor. There is an ultimate common ancestor that exists that was the beginning of all living things. It's just the proximity to the current time that determines how closely related we are to something. So like we have a common ancestor with an alligator, but that's going to be way further back than our common ancestor with a monkey, etc. Okay, so practice tests do not use anything before 2013. So 2013 to 2018 is where you want to live for practice tests and practice free responses. Sorry, just reading that over there on the side. Okay. So mitosis and meiosis, I have a great song that some of you I'm sure have heard maybe now multiple times, and it's mitosis is in mitoses, meiosis is in meioves, and that helps you remember that mitosis is in body cells and meiosis is in ovaries, so sex cells, so it's going to make our egg and sperm. General things, you do not need to know PMAT, you do not need to know prophase, metaphase, anaphase, telophase. You just need to know that mitosis, we're doubling our DNA and then dividing one time. Our daughter cells are identical. Crossing over does not exist because we are not lining up by homologous chromosomes. Let's see what else. It's all highly regulated. So P53 is a tumor suppressor protein. So it just helps regulate the cell cycle. And if it sees rapid growth or that cells are going through checkpoints when they shouldn't be, it will go ahead and kill those cells through apoptosis. Whereas meiosis is going to end up, we duplicate our DNA once, divide twice. It's going to end up in 4 genetically unique daughter cells. Crossing over does occur because we do line up homologous chromosomes and we get a lot of genetic variation from that. And all of our daughter cells have half the amount of DNA as the original cell. So that's my real quick crash course, but I do go through a lot of that. You do not need to know prophase, metaphase, anaphase, telophase. They removed that. If you actually take a look at that massive PDF that I sent you earlier, it specifically does say that in there. You just need to know that chromosomes are lining up and then being pulled apart. And that's to make sure that we get the right amount of chromosomes on either side of the cell. All right. I would not think that you would ever need to know that P53 is a tumor suppressor protein. Good question, Ria. If I had to guess, if they told you about it, they would tell you all the information about it, and they would just need you to apply it to a situation. That's usually how things like that work. So if you know it going into it, that's wonderful because you'll feel more comfortable. But if you don't, you know, no sweat, you'll still be able to answer the question. Okay. Can you go over a population math problem? Sure. Let me see. I had some examples. Let me pull them up. Oh my gosh, this might be my last question that I'm able to answer. Let me see if there's any other quick ones before I go through all the thing of pulling this up. Oh, I know exactly what question you're talking about, and they would accept mutualism as well. It's the bacteria question I'm assuming you're talking about, or how do bacteria differ from the inside versus the outside? Mutualism is an acceptable answer choice there. You do not need to know too much about rhizobium and nitrogen fixing. You should know that there's nitrogen fixing bacteria that are going to fix nitrogen and make it usable for plants, but plants are a very, very minimal part of the AP exam, so don't stress too much about them. You just need to know that bacteria are going to take nitrogen out of the air and make it usable to put into our organic compounds. I am going to hold off on that ecology practice problem, but come back during a cram session or look up ecology practice problems just because I don't want to take time away by trying to find my PowerPoint slide and going through that right now, so I can get through as much as possible. Biggest topic on the exam is definitely evolution. Sorry, I just saw that on the right-hand side. Okay. Multiple choice for each unit. I do have some practice ones that I can post. I really like Albert's multiple choice questions, although some of them aren't as rigorous as the AP exam. Unfortunately, most practice tests are under lock and key, so you can ask your teacher. That's my best guess of how you can get access. So like I have a ton of practice tests, but I can't post them for you because if the answers end up online, I lose my ability to teach an AP class. But your teacher can print them for you and you can practice them in their classroom under their supervision. So that would be my best suggestion for you. But there's a lot of study books out there. Cliff's Notes is one that I use in my class that have tons and tons and tons of practice questions that are pretty affordable that you can real quick Amazon Prime. But yeah, or ask if you really want the real deal, because no practice test is as good as the real deal. It's just not, because it's not created by College Board. Ask your teacher if they'll print a practice test for you and you can practice in their classroom. Okay, what kind of immune system, nervous system, and body system should we know or expect on the test? You need to know endocrine, nervous, and immune. That's it. Those are the only three that you need to know. Sometimes circulatory shows up, but they give you all the information in advance. You do not need to come in with that knowledge. So for immune system, you should know the difference between non-specific and specific, first, second, third line of defense. You should know B cells and T cells. You should know some examples of non-specific responses. For nervous system, you should be very familiar with the action potential and the neuron and what happens at a synapse with neurotransmitters, because I've seen a lot of free response lookup. AP Biology Free Response 2016, because that's a really good example with a scoring guideline of what you need to know for the body systems. Okay, does anyone know a good website to review? Okay, so yeah, it looks like a lot of people have already answered this question, so I'm going to leave it to them. One of the future videos, please talk about the properties of cellular components in DNA. Sure, I can definitely go through some of those. I talk about them. I have a biochemistry podcast that I would, or screencast, not podcast, that you can watch that I would highly recommend viewing. But I can certainly go over like which atoms are in which macromolecules. Okay. What would I say is the hardest part of the exam? Time management. 150%, it's just going to be managing your time. You're AP students, so you're like me, you're very, very stubborn. And so you're going to want to stop and answer every single question, but you're doing yourself a major disservice if you try to do that. So the biggest thing to do is swallow your pride and skip questions if they're taking you too long, and come back to them if you have time. If you have time, then what's the harm in skipping it in the moment? But the hurt is major, because you're not going to get to all the questions, and then you're going to miss out on a lot of possible points. I think I told you guys this last week, but multiple choice go from easy to medium to hard to medium to easy. So if you're not getting to those end questions, you're missing out on a lot of free points. So that truly is the hardest thing. It's also why I tell you to start with the short free response and move your way up to the long free response, because it is very easy to get yourself lost in a 45-minute writing spree for question 1 and then have absolutely zero time for questions 6, 7, and 8 when you probably would get more points on those ones. Most students do not do too well on the long free response. The more you write, the worse students actually usually do. So I would say limit yourself to like a 1-page response for the long free response. Ooh, chi-square. Wow, such a good question. Wish I had time to go through it. I'll make that a priority for our cram sessions. Sarah, hopefully I answered this one. Okay. Hopefully I also answered the commensalism one. I can give a couple examples there, or you can Google it. When predators are high, what is their prey? I'm confused of this question. Their prey is still their prey, but usually the prey then starts to decrease because there's lots of predators. And then once prey starts to decrease, predators will eventually follow because they run out of food. Okay. Oh, I know. seconds. Ooh, wow. Okay, Savannah, that's a great question. I'm going to answer that really briefly. I would put money on the fact that there's going to be a chi-square question on your test. I don't know it for sure. If I did, I would tell you everything, because that would be great. You'd all pass with flying colors. But there has not been a free response since 2013 that has chi-squared, so I'm just like betting on it happening this year. Okay. Ooh, old videos of me. You get a Fiveable Plus membership at the Fiveable website. So go to the Fiveable website, get a Fiveable Plus membership. You'll enter into my class, and everything ever that I've ever done ever is in there for you. Yay. Ria, let me try to answer this really briefly. So B cells produce antibodies and bind to pathogen that is free floating in the blood. T cells are going to attack cells that are infected, so they contact cells, not free pathogen, or they bind up with antigen presenting cells. And helper T cells are the ones that meet up with an antigen presenting cell, such as a B cell or a macrophage, and send out a signal that activates more B cells and more T cells to the area and tells us that we're under attack. Yeah, you had to do an FRQ. If you look at 2013's question 1 for chi-square practice, yeah, there it is. The first one of 2013 is going to be a chi-square practice. Okay, perfect. All right, y'all. The only thing that I could not figure out is how to post this document, and I'm really upset about it. So I will figure out a way and make sure that it gets dropped into, oh, you know what I'll do? I'll just have it dropped into my folder. I'll upload it into my folder so that you guys have access to it if you have a Fiveable Plus membership with all of my grid-ins. You're right, these do pass too quickly, which is why, so all those documents will be available to you if you have a Fiveable Plus membership, and I will make sure that that one goes up in there so that you guys have those practice problems. Okay, so reminder, and this is the deal of the century, it's $5 to have access to my next 3 review sessions. I promise I will have more time to answer all of your questions, for sure. And yeah, so come back. Unfortunately, we run out of time. It's not on Google Docs. Otherwise, I would just share it with you guys. It's in Word. I don't use Google Docs because I hate it, which is why I was trying to convert it into a Google Doc, but it didn't work. Yeah, I'm sure you can rip your appendix sheet out. I don't see why not. That's another good question though. All right, I'm gonna hop off. If you still have like a few questions that I can answer with a quick word or two, you can put it in the chat box. I know we were all over the place tonight. Hopefully you gained something from it. Again, I'll try to answer even more of your questions from over the next 3 days, Friday, Saturday, Sunday. So definitely stay tuned, and I can go over more testing strategies as well. All right, guys, it's been real. See you on Friday, hopefully. Get that Fiveable Plus membership. Bring your friends.